Good morning, children. We have started with 6th chapter, Molecular Basis of Inheritance. Today also we'll continue the same. The topic which we are going to start today is transcription. It is the process of copying genetic information from one strand of DNA into RNA. Okay, do you remember? Last time also I have told you that the process of copying DNA information which is present on DNA into RNA is known as transcription. Also, the process of transcription is governed by the principle of complementarity, except the adenosine now forms base pair with uracil instead of thymine, because thymine is not present in RNA. Unlike in replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA or only one of the two strands is copied into RNA. Reasons why both the strands are not copied at the same time during transcription are, number one is, if both the strands are copied, they would code because for RNA molecules with different sequences, and because complementary does not mean identical, and in turn two proteins with different amino acid sequence would be formed. Hence one segment of DNA would be coding for two different proteins, and the genetic information transfer machinery would become much more complicated. So this is one reason. That is, if both the strands of DNA are transcribed, two different types of proteins will be formed. Okay, and it would be that DNA would be coding for two different proteins, and the genetic information transfer machinery would become more complex or complicated. The second reason is the two RNA molecules produced simultaneously would be complementary, because the DNA strands are complementary to each other, both the strands. So the two RNA strands which will be produced from the DNA will be complementary to each other, and hence would bind together to form double-stranded RNA. And this would prevent RNA from being translated into proteins. Okay, so if two strands are produced, both the strands will be complementary and they will form the double-stranded RNA. And if double-stranded RNA is formed, there will be no translation process taking place, and if no translation, then the proteins will not be formed. Okay, now we will move to the next topic, that is a transcription unit. A transcription unit of DNA is defined primarily by three regions in the DNA. And the three regions are a promoter, the structural gene, and a terminator. The two strands of DNA have opposite polarity, and the enzyme DNA dependent RNA polymerase catalyzes the polymerization in only one direction, that is 5' to 3' direction. The strand with 3' to 5' polarity act as the template strand and is known as the template strand. The strand with 5' to 3' polarity and same sequence as RNA, except thymine at the place of uracil, is displaced during transcription and is known as the coding strand. Children, I hope this is clear. Okay, the strand which has 3' to 5' polarity. This is called the template strand. And this strand will get transcribed into RNA. Okay, and the other strand that is having the polarity 5' to 3', which will have the same sequence as RNA, except thymine at the place of uracil is displaced during the transcription process, and this process is known as the coding strand. Okay. Now, this is the schematic structure of a transcription unit, where you can find the promoter region. Here you can see promoter region is there. Okay. Then this is the transcription unit. Okay, this is called transcription unit, or you can say this is the template strand. And this is the coding strand. At the end, the terminator is present over here, which will terminate the transcription process. So let us study about all these units. A promoter, is a DNA sequence that provides binding site for RNA polymerase. Okay, that means RNA polymerase will bind to first which region of template strand? That is promoter. It is located at the 5' end upstream of the structural gene. Its presence also defines the template and coding strands. The definition of coding and template strand could be reversed by switching the position of promoter and terminator. Then the structural gene. The structural gene is a transcription unit. Okay, that structural gene in a transcription unit is flanked by the promoter and terminator. And the third one is the terminator. A terminator is located towards the 3' end downstream of the coding strand. It usually defines the end of transcription process. Okay. Now let us study about transcription unit and the gene. Gene is the functional unit of inheritance. The DNA sequence coding for tRNA or rRNA molecule also defines a gene. Second is cistron. Cistron is a segment of DNA that codes for a polypeptide. In a transcription unit, the structural gene could be monocistronic, mostly in eukaryotes, or it may be polycistronic, which is mostly in bacteria or prokaryotes. The monocistronic structural genes in eukaryotes have interrupted coding sequences, and they are of two types, that is exons, coding sequences or expressed sequences that appear in mature or processed RNA, and introns, that are intervening sequences which do not appear in mature or processed RNA. They only interrupt exons. The split gene, that is gene with both exon and intron, is a characteristic of eukaryotic DNA. The promoter and regulatory sequences of a structural gene also affect the inheritance of a character. That is why sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. Okay, now let us study about transcription in prokaryotes. DNA-dependent RNA polymerase is the single enzyme that catalyzes the transcription of all types of bacterial RNA, as shown in the figure, and takes place in 3 steps. The first step is initiation, then elongation and termination. Let us study about the first step, that is initiation. RNA polymerase binds to promoter and initiates transcription by associating transiently with initiation factor rho. Sigma. So sigma factor will get associated with RNA polymerase and it will then bind to the promoter region as shown in the diagram. It uses nucleoside triphosphates as substrate and polymerizes in a template-dependent fashion, obeying the rule of complementarity. It also facilitates opening of the helix and continues elongation. Then the second step The second step is elongation. Chain elongation proceeds in 5' to 3' direction and the transcription bubble travels with the RNA polymerase. This is transcription bubble. Right, so it travels with the RNA polymerase. The RNA polymerase, after initiation of transcription, loses the sigma factor. You can see here the sigma factor is being lost, but continues the polymerization of ribonucleotides to form RNA. Okay, so sigma factor will get detached from the RNA polymerase and the RNA polymerase will continue the polymerization of the ribonucleotides, which will result in the formation of RNA. Here you can see the blue color line. That is what? RNA. So RNA will be formed. Now let us study the last step, and that is termination. When the RNA polymerase reaches the terminator region of DNA, okay, when you can see here the terminator region is present, right? So when the RNA polymerase reaches to the terminator region of DNA, which is GC rich, guanine and cytosine rich region, and has a hairpin like structure, the nascent RNA separates, means the RNA which is formed gets separated and the RNA polymerase falls off. You can see the RNA polymerase gets separated, even the RNA strand which is being synthesized is also separated. So first the RNA strand which is synthesized will get separated and there will be separation of the RNA polymerase, resulting in the termination of transcription. The RNA polymerase is only capable of catalyzing the process of elongation, but it associates transiently with initiation factor sigma and termination factor rho to alter its own specificity to initiate and terminate the transcription respectively. Okay, so during termination, the RNA polymerase will get associated with the rho factor, and during initiation it will get associated with the sigma factor. In prokaryotes, mRNA does not require any processing to become active, and both transcription and translation takes place in same compartment, as there is no separation of nucleus and cytosol in bacteria. Therefore, translation can start much before the mRNA is fully transcribed. That is, transcription and translation are coupled. This occurs only in prokaryotes, that is in bacteria. Okay. I hope all these 3 steps of transcription are clear to everyone. That is, initiation, where the RNA polymerase gets attached to the promoter region associated with the sigma factor. Then elongation, there will be transcription bubble will be formed, and the RNA polymerase will proceed in the 5' to 3' direction, and it will get dissociated from the sigma factor, and it will continue the polymerization of ribonucleotides and will result in the formation of RNA. And the last step, termination, when the RNA polymerase reaches the terminator region of DNA, which is rich with GC, there will be association of RNA polymerase with the rho factor, and the RNA strand which is synthesized will get separated, and after that the RNA polymerase also gets detached, which will terminate the process of transcription. And the process of transcription in prokaryotes takes place in the transcription and translation takes place in the same compartment. Before the mRNA is fully transcribed, translation process also starts. Okay, so that is all about transcription in prokaryotes. Now we will study about transcription in eukaryotes. The process of transcription in eukaryotes is similar to that in prokaryotes. Structural genes are monocistronic in eukaryotes. 2 additional complexities are present in eukaryotes, like number 1, in nucleus there are at least 3 RNA polymerases. In addition to RNA polymerase found in organelles to allow clear-cut division of labor, like RNA polymerase 1, it transcribes rRNAs. rRNAs are the ribosomal RNAs. Okay, so RNA polymerase 1 transcribes rRNAs 28S, 18S and 5.8S. Okay, then the second type of RNA polymerase is the RNA polymerase 2. It transcribes precursor of mRNA, which is called heterogeneous RNA. It is called heterogeneous RNA. So the precursor of mRNA, which is called heterogeneous RNA, is being transcribed by heterogeneous nuclear RNA. Heterogeneous nuclear RNA. So heterogeneous H N nuclear RNA. So heterogeneous nuclear RNA is being transcribed by RNA polymerase 2, and the third one is RNA polymerase 3, which transcribes tRNA, transfer RNA. Phi srRNA is also being transcribed. srRNA and snRNAs are also being transcribed, that are small nuclear RNAs are also being transcribed by RNA polymerase 3. Then the primary transcript which is being produced contains both exons and introns and are non-functional. The primary transcript which is produced, which contains exons and introns, they are non-functional. Therefore, they undergo a process called splicing to remove the introns and to join the exons in a proper order to allow translation to take place. In presence of introns in reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. The heterogeneous nuclear RNA undergoes two additional processes, that is post-transcription modification. Okay. There will be post-transcription modification taking place. Okay, now suppose exons these are exons, okay. Then introns are also present. This is heterogeneous nuclear RNA which is being formed. Okay. Now there will be this is 5' end and this is 3' end. So there will be addition of triphosphate methyl guanine at the 5' end, and at the 3' end there will be polyadenylation or you can say tailing takes place. Okay. Then there will be removal of this introns will be removed, right, and the exons which are present, which are there, will form a continuous strand, and this is what? This is the messenger RNA. This was hnRNA, heterogeneous nuclear RNA, which contains exons and introns. Okay. So this process of removal of introns is known as splicing. So children, I hope this is clear, right? So capping takes place, as I have explained you. In capping, methyl guanosine triphosphate, an unusual nucleotide, is added to the 5' end of hnRNA, and tailing process also takes place. In tailing, adenylate residues, that is 200 to 300 adenylate residues, are added at 3' end in a template independent manner. It is the fully processed hnRNA, which is now called mRNA, and gets transported out of the nucleus for translation. So children, I hope you have understood that in prokaryotes, translation and transcription and translation occurs in the same compartment, whereas in eukaryotes, transcription occurs in nucleus after the formation of mRNA. The mRNA gets transported in the cytoplasm, and in the cytoplasm translation takes place. Okay, so that is all for today. In our next video, we will be studying about genetic code and mutation. I hope you all have understood this. Thank you.